Okay.
And stand close to Parul so people can kind of hear you.
Okay, so people online can kind of hear me. Here, I'll get on this side of you.
That should be fine.
Hello, everybody. Hello, people in Zoom land. It's my great pleasure to introduce Parul Johri, who's come all the way here from North Carolina. Let's see. So Parul did her PhD with Mike Lynch at Indiana, where she did some really interesting work looking at ciliates, actually, now that I think back on your PhD thesis. And then for the last few years has been in a postdoctoral position with Jeff Jensen at ASU. I could say there are a couple things that I'll mention. The first thing that I mentioned is one thing that I really admire about Parul's research program, writ large through her PhD and her postdoc, is she's had a laser-like focus on the fundamental forces that drive population genetic variation. So like mutation, recombination, sort of gritty, bare bones stuff. And it's super important. It's a super important line of research. The other thing that I'll mention just briefly is that Parul has just started her own new lab at UNC and is looking for recruits, PhD students, and postdocs. So I'll pre-advertise for you. Anyway, I'm really excited for this talk. Welcome, welcome. Such a pleasure that you're here.
Thank you so much, Andy. Thank you so much for the invite and for that introduction too. I've had the pleasure of talking to many people here today. It was really great, really fantastic. So thank you for taking the time out to talk to me. Today, so let's get started. And as Andy said, I've been a postdoc. Oh. Right. Okay, so this is gonna take. All right, let's see if this is gonna work.
It's working.
Is it, yeah? All right.
Well, sorry, it looks good, but it's the presented mode, right? We are seeing your laptop screen and not the projector.
Right.
Oh, wait, no, but now.
Yeah, does that work?
I don't know.
Oh, how do I?
It looks good, I think. Okay, let me move that's not on. Is that the, you're just kind of.
Yeah, that's it.
A little bit?
Oh, really?
No, that's good, yeah.
I think that's good.
Everything. Excellent.
Okay, all right. Well, finally, we are here. Okay, thank you for bearing with me on that. Okay, so what I want to talk to you about today is the role of non-adaptive processes in shaping genomic variation. So we know that when we can look. Sorry about that. Got to figure out how to. All right. We know that when we get information about genetic diversity or genetic variation information from a natural population, it can yield lots of interesting insights. One of them, for instance, is about their past or historical population size changes that the population has undergone. So for instance, if you have sequences from multiple humans, we can put together the historical events of how and when they migrated across the planet. And so, these kinds of inferences, they're important to understand how genetic diversity is sort of shaped today in the world. And this is one example. You can also understand evolution of pathogenic species inside a host, et cetera. So there are many different things you can do with when you have genetic variation data. And of course, to really understand how this kind of evolution is occurring or to make such kind of predictions or inferences, right, we need to understand how evolutionary processes really shape genetic variation. So the evolutionary processes like Andy just mentioned that I'm really referring to are things like selection, mutation, recombination, and population size changes. And all of these together determine patterns of variation. And what we actually do most of the time is in a population, we usually take a snapshot at any time point. And what we're really asking is, okay, what are the, how have these processes historically shaped variation up to that point? Can we make those predictions? So I know this will be a very basic for a lot of people, but I think that just quickly reviewing what I mean by all of these evolutionary forces and how they impact genetic variation, mutation, for instance, is basically a major source or primary source of new variation because it adds new alleles to population, right? And recombination, what it does is it also adds new variation to population because it's important for reshuffling alleles and producing new allelic combinations, right, in the population. And then you have selection, right, where when you have selection acting on a particular allele, for example, if it's deleterious, it can disappear from the population. If it's beneficial, it can increase in frequency. And so you're able to change allele frequency of alleles because of selection. And same thing can happen with demographic history, where, for example, if you have a severe bottleneck, you can kind of lose high frequency alleles and lose variation. So the main point of this slide that I want to communicate is that all of these evolutionary processes together generate patterns of genetic variation, and it's sort of difficult to disentangle their effects sometimes. And that's because a lot of times they affect variation in a similar way. And this led to a lot of sort of fundamental debate in the field for a while, because about asking whether it's for example, adaptive processes like positive selection that's largely shaping genome-wide patterns of variation, or if it's a combination of all the other non-adaptive processes. And this debate has been on for a while, actually, starting from Wright and Fisher, then going on to Kimura and Ota and Gillespie. And, a large part of this debate for a while was restricted to sort of functional parts of the genome, and now has sort of taken a different form where it's important for sort of genome-wide variation, where it's not just restricted to functional regions. And why is that the case? That's because while things like population size changes can, affect variation at genome-wide level, so can selection. And so here I'm just showing you a very quick example where these blocks, these functional blocks, they represent some sort of functional part of the genome, which is important. And so if you look on this side, if you have a beneficial mutation at that part and it increases in frequency, of course, other alleles that are linked to it can also increase in frequency, even when they don't contribute to fitness, right? And in a similar way, if you have a deleterious mutation and it gets purged from the population, it will take with it other neutral alleles with it. And so what can happen is that selection can actually start affecting other parts of the genome, even though they're not directly impacting fitness. And so this is one of the main reasons why it's been difficult to disentangle all of these evolutionary processes from each other. So some of the questions that have sort of driven my research are, what are the consequences of linked effects of selection across the genome? And how can we disentangle these evolutionary processes from each other using population genetic data? And what is the role of adaptive versus non-adaptive processes in shaping genome-wide variation? And we think that, being able to answer this question, it's been difficult. And part of the reason is that we want to start having the appropriate sort of null model or baseline model for being able to ask these questions. And by that, I mean that we want to start accounting for processes that are constantly occurring in populations. So for instance, something I didn't tell you about is that whenever there are new mutations in populations, a majority of them are actually deleterious or effectively neutral. So there's a pretty large mode at the point of effectively neutral or sort of weakly deleterious mutations. And then there's also usually we now think it's almost 50% sometimes of mutations that are very strongly deleterious. And beneficial mutations are a smaller minority of new mutations. So, these effects of background selection and purifying selection are sort of pervasive in populations and are constantly occurring. And on top of that, natural populations are always undergoing a change in population size. They always fluctuate, and there's no reason to believe that they're under equilibrium. And so the demographic history and sort of direct and indirect effects of purifying selection, as these are sort of constantly acting in populations, it's important to start accounting for those as your as your baseline model. And so what I want to talk to you about today is I want to talk a little bit about the effects of that deleterious mutations have and the consequences of not accounting for them in evolutionary inference. And then I want to talk about this in a slightly larger perspective of simply problems with model fitting in population genetics whenever we perform statistical inference. And then I want to take you through our computational solution and its application and implications. So, right, so we've sort of already touched upon this, that so this process is actually called background selection, where if you have a deleterious mutation at a functionally important site, it can change or decrease variation at the linked neutral sites, right? And this process has been studied pretty well for a long time, actually. And we have a decently good idea about sort of the theoretical underpinnings of this process. And it's been able to be modeled pretty well. Like if you have a functional site, you can start predicting what would be the decrease in variation at sort of a linked neutral allele from that. And one of the main things basically to note here is that, as you might expect, it's directly proportional to the mutation rate, like the deleterious mutation rate. So more deleterious mutations, more is the effect. And it's sort of inversely proportional to the recombination rate because you're breaking up linkage in that case. But the important part is that it's a much more complex function of the selection coefficient. And so the effects of background selection, they maximize at a specific distance from, depending on the selective effects. And so all I'm trying to say here is that it's important, the entire distribution of fitness effects is what determines how background selection effects occur around a functional element. And so this is just something that, so basically, if you really want to describe these effects, you have to integrate it over the entire functional site and all the selection coefficients. And this is sort of just an aside, but we noticed that when you have to do this, you usually have to numerically integrate over this because your distribution of fitness effects can be a complex function. But we went ahead with making it into a simpler format and just discretized it. And that allowed us to get analytical expressions for these effects, which can be helpful sometime downstream. And so what we did is, this is sort of a discrete distribution of fitness effects where we broke it up into 4 classes, where the first one is effectively neutral, the second is weakly deleterious, and then moderately deleterious, and then the 4th class is very strongly deleterious. And because we assume that these are uniform functions, so it's actually really easy to get analytical expressions for that. And we're able to predict the recovery of this diversity away from functional elements pretty accurately based on this analytical solution. But here's the thing that, this is the work that we have known for a while, that background selection decreases variation, and you can sort of predict pretty well. But there's another effect that background selection has, which is much less predictable, which is that it actually changes the entire distribution of allele frequencies of nearby linked alleles. And so here, I'm showing you sort of a distribution of derived allele frequencies. And so this just tells you, for example, that alleles at 0 or 0.1 frequency in the population, like about 70% of those alleles are at 0 to 0.1 frequency in the population. And the black line, the black bars show you that for a neutral equilibrium scenario. And the red bars show you that for, a scenario where you have equilibrium, but you also you're counting for effects of deleterious mutations. So what I'm saying is that background selection causes this sort of skew towards rarer alleles at linked neutral sites. And when we usually do population genetic inference, we're sort of focusing on, we always assume that it's strict neutrality. And so we're assuming it's this black distribution. And of course, there are other processes, other neutral processes that would similarly shape the site frequency spectrum. For example, if I add in exponential growth, it looks very similar to background selection effects, which is shown in the green bars. And so you can see it's going to be difficult to distinguish between exponential growth and background selection. And so just to sort of look at this in a context of the methods that we use today, and sort of at genome scale levels, we sort of simulated somewhat realistic genomes where we sort of mimicked a 150, like we simulated 150 MB chromosome regions from the human genome. And we sort of had an intron exon intergenic structure, and these exons were experiencing selection, in this case purifying selection only, and everything else was neutral, which we used for inference. And you see there was sometimes a pretty drastic misinference effect, where the black line here is telling you what the true population history was. And note the time goes from like past to present. So this is actually a decline in population. And when we use standard methods, which are shown in these colorful lines, we instead infer a very recent growth. So it really gets it very wrong in certain cases. It does depend, though, it doesn't always get it wrong. It depends a lot on the demographic history. So in this case, under equilibrium, the misinference is a little bit lesser. When you actually, if there's true growth, then there's no misinference, and that makes sense. With decline, it's the most misinference that you can get. And of course, as one might also predict, it depends on the density of selected sites in the genome. So, based on what we simulated, if 5% or 10% of the genome is under selection, you don't really have any misinference, which is the case for human genomes. And so actually, it doesn't really have much implications for actual human demographic history. But we were interested in knowing what would happen if you do have these kinds of scenarios. And so if it's above 20% here, you have pretty drastic misinference. And why this is relevant is that we don't apply these methods only to humans, right? We also apply them to many, many organisms, and they have been applied to many organisms. And you don't account for their like density of selected sites in the genome. And that varies quite a bit also. So, humans is on the lower end, but then here I'm just kind of showing you a bunch of model organisms, and, Arabidopsis and C. elegans is like about 30%, Drosophila and yeast 50 to 60%, unicellular eukaryotes go up to like 60 to 80%, and then viruses, of course, 90%. Like SARS-CoV-2 is actually about 99% coding. So, it's a question like how accurate are these methods in these other organisms that have so much higher density of selected sites. So summarizing this first part, I've shown you that segregating deleterious mutations affect variation at nearby sites, and if we don't account for them, we can lead to misinference of population history. And as I was saying, this isn't really a problem restricted to just background selection or demographic inference. This is really somewhat of a problem at a larger scale when we perform statistical inference. And this is because, as I've kind of already said before, a lot of, all of these evolutionary processes are somewhat affecting or getting information from similar pieces of information. And so, for example, we know that you need to account for demographic history if you want to infer parameters of selection. We also know now that you kind of need to account for selection to be able to get accurate demographic history. We've always known that, mutation rates you have to know them to actually know the population sizes. And we also know that we're starting to know that recombination and gene conversion rates are important for getting accurate demographic history estimates, and of course, for selection. And then, there's more recent work that suggests that if you don't account for demographic history, you might actually misinfer recombination hotspots. And so there's this whole issue, right, where everything is connected, but, and so if you're starting on sort of a new organism or a new analysis, there's a problem with where you should really start, because you end up with this sort of circular problem. And on top of this, there can be this issue that, maybe some of these evolutionary processes together could possibly explain the data equally well. And so then, where would you go from there? How would we know what's really going on? And just to sort of formalize that a little bit better, we were interested in trying to see if we can fit really two very unrealistic and not unrealistic, but two extreme scenarios that we know might be incorrect in a natural population. So what we did is, so we went with two models that are sort of on the two extremes. One, which is that everything is strict neutrality and you're just fitting everything to a demographic model. And one where you're assuming that all of the genetic variation is explained by positive selection. And so in the case of neutrality, we just fitted like 3 parameters. So it was a simple model. And in terms of positive selection, we fitted the proportion of new mutations that are beneficial and what is their mean selective effect. And we modeled it as an exponential distribution of fitness effects with like mean S. And so as one might predict right now, if you have a population that has deleterious mutations in it, which we know they do, and that's how they evolve, we know that we can, actually, just a quick explanation. What you're seeing here is we fitted these models here, right? And so what I'm showing you here is like a joint posterior. So here are the 2 parameters for the demographic history, and here are the 2 parameters for positive selection. The red cross tells you what the truth is, and the black posteriors are what we infer. And so you can explain the data by sort of a mild growth, or you could explain it by sort of weak and infrequent recurrent sweeps. And so it will be hard to distinguish these 2 data from each other. And in of course, if you add in more things, now this is a population where more of it, everything is going on. There are beneficial and deleterious mutations and their population sizes. Again, now what we see, again, is that, you can explain the data by sort of this very, very strong recent growth, or you could explain it by very infrequent and very strong recurrent sweeps. And so, just coming back to the problem of how it's hard or it's difficult to be able to distinguish what's actually going on in these scenarios. And so what's our solution to this problem? Well, one possible solution that we have tried is to start to build this so-called null or baseline model where we are accounting for demographic population history jointly with direct and indirect effects of purifying selection, and then being able to perform joint inference of that in a context where you're accounting for these linkage effects. And so that's the next part of this is what we next part telling you about what we did here and what we find when we applied this method. And so what we really did here is we simply considered particular functional regions with particular lengths and their recombination and mutation rates across, say, like the Drosophila genome. So we applied it to Drosophila and humans. So, we used different sections of these genomes. And we simulated a large number of different distribution of fitness effects and single size change in population history. And the idea was that we sort of wanted to use the decay of these background selection effects around functional elements to be able to get information about all of these parameters and jointly infer them. And it turns out that we did pretty well. Here is the inference for all of the DFE parameter estimates on top, and the two for demographic history. So we seem to be able to infer these parameters jointly pretty well. This was done in an ABC framework for people who want to know more, and the simulations were done in SLIM, so it accounts for the linkage effects in these cases. And this is just sort of an interesting thing, but we found that if you actually only use just the functional region and don't really think about the decay effects, you still can infer all of these parameters, but you can see there's an increase in error, of course, in these cases. But the reason this is useful, and I'm mentioning it, is only because it can be helpful for really sort of like viral genomes, which are really 90% coding or 99% coding, and you have no idea what's functional and what's not functional. We've applied it to human cytomegalovirus and influenza virus, and we're now working on applying it to SARS-CoV-2. So let's see, hopefully we can find something interesting. So yeah, just quickly, I want to tell you about why, like what aspects of our framework were novel here. So we can fit multiple aspects of the data. So you can account for allele frequency distribution and association between alleles, like linkage disequilibrium, and also divergence from sister species. So we were fitting all of these statistics to infer these parameters. And of course, we can account for linked effects of deleterious mutations. And there were no assumptions made about which of the sites were neutral within the functional region. And so there's some advantages of that, like we can use exon data for inference, or we can really infer the DFE of regulatory mutations because we don't really need synonymous sites in that case. Oh yeah, and we can use it in non-model organisms where we don't really know, for example, which sites are actually important and which ones are not. So you can just infer the DFE of the entire functional region. So, and lastly, we also make no assumptions about selection on synonymous sites. And that was, so if you do have an organism where synonymous sites could be under selection, this could be an important sort of factor that would bias that inference. So currently, when we normally perform DFE inference, it's sort of like a two-step approach that we follow, where we use synonymous sites from a functional region. We assume they're neutral. We infer the demographic history, and then we take the non-synonymous sites and we condition it on the same demographic history inferred from synonymous sites, and then we can infer the distribution of fitness effects at these non-synonymous sites. But the idea is if the synonymous sites are not neutral, your demographic history won't be accurate, and so you'll have downstream effects. And we tested that by sort of simulating 30% of our synonymous sites as experiencing weak selection. And here, I'm going to show you what was the truth and what was our inference. And we used a sort of standard approach here called DFE alpha that infers fitness effects in this way. And so we found that the pattern bar is that DFE alpha, and so it tends to underestimate basically the selection that's experienced by the synonymous sites. Like if it's weak selection, it underestimates that. And of course, it'll estimate a growth because that's what selection would do. So that's not surprising. But we do find that it's possible that you may not uncover or see these weakly selected mutations. So, and the blue bars show you our inference, and I just want to point out that works very well in this scenario. And so we went ahead and applied this to Drosophila data. This is from the Zambian population. And here what I'm showing you, okay, so the upper panel shows you the demographic history, again, past from present. All the colorful lines are showing you like previous inferences, and the black lines show you our inference. And so earlier it was predicted that this population has likely had recent growth in this population, but our method actually says that's almost no growth or close to equilibrium. And I just want to point out that that's what one would expect if you were not accounting for background selection. You would infer recent growth, and we don't. So I think it's potentially possible that after our method is able to account for these background selection effects. And then at the lower panel is the distribution of fitness effects. Again, the colorful lines are telling you what was previously there, black lines for us. And we actually infer a very large class of mildly deleterious mutations as compared to what was inferred previously. So, yeah. And we've also recently performed this inference for the human Yoruban population. And here actually our results are sort of different, which is that they're very much in line with what other previous estimates were. So it seems like the black line is kind of approximately similar to what the other estimates were previously. And with the distribution of fitness effects, it follows this, it's very similar to this yellow bars that you see, which is the most recent inference of population of the distribution of fitness effects by Huber et al. Done in the same population. And so it seems like basically the humans have a lot of, effectively neutral and mildly deleterious mutations, and it seems consistent with other previous studies. And it makes sense too, because in humans, you're gonna have, like I showed you earlier, the density of sites is lower. So actually our joint inference probably hasn't made a big difference as compared to what happened in Drosophila. Yeah, and the good thing is that we were also able to look at so after you take your best model, you can ask, does it fit the data that you're observing? And so this is actually for the human population. I'm going to just keep going back and forth because we find very similar results with human and Drosophila. And so this is the distribution for all of the exons that we chose. So this is like the distribution of per exon information. And this is, this is the functional site, and this is the window next to it, which is linked, and then the one that's less linked. And we're able to so okay, the white distribution, or the one with the black lines, is the data, and the red is the best model that we have inferred and simulated. So it's explaining the data pretty well here. There's like the top one is for a summary of the site frequency spectrum. The second one is a summary of the linkage disequilibrium, and the last one is divergence. So we're able to explain different aspects of the data. Yeah. 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 This is just once you've inferred the best model, are you still able to explain it? Like, are you just not randomly finding, you've inferred such a weird thing that you found something, but it doesn't actually explain the data? This is just a double check to see, yes, we're explaining it. Okay, yeah, sure. But I don't think I'm just saying that the best model that we infer is explaining the data, unlike in cases where you don't. Okay. All right, so we then next asked, what about, we didn't consider, the presence of beneficial mutations in this model. And so we were wondering what would happen if we were to simulate that on top of this. I mean, how would positive selection affect, these fits? And so what we sort of did is we just sort of simulated in sort of a grid where we had, we had different frequency of new beneficial mutations, which is going from like 0.1, 1%, and 5%, and then different strengths of selection. Again, this was exponentially distributed with this mean. So, and the idea being that here you're kind of looking at common and strong positive selection, and here you're looking at rare and weak positive selection. And it turns out that in both of the Drosophila and human cases, the fits are not very consistent with very common and strong positive selection, but they're extremely consistent with the moderate to weak. And I mean, I'm using the word rare, but these are all rare. So that's not really a way to say it, but between 0.1 to 1%, they were always consistent with the data set. And this is just an example of like haplotype diversity that was seen in the Drosophila population, where, for example, when it's weak, the distributions are pretty overlapping when you have selection, and when you don't, those are the two gray bars. And in the case where you have very strong selection, the distributions are pretty different. And so in that case, it's kind of obvious you're able to identify that on top of each other. So yeah, so what we think is that positive selection is probably likely not very strong and frequent, by which we just mean 5% in Drosophila in humans, and that it doesn't improve the fit of the best model in humans, but not in Drosophila, and suggests difficulty in identifying these parameters of sort of recurrent positive selection if we were to try that from human population data. And that joint inference of selection and demography will be more important in organisms with high density of selected sites. Like it didn't tell us much new stuff in human populations, but it did tell us more things in the Drosophila populations. And especially important for organisms that have low recombination rates, or generally non-model organisms where you don't know what is functional. So I want to quickly go over two sort of population genetic implications of our work. One is that we found the presence of more weakly deleterious mutations than was previously found in Drosophila. And it has some interesting implications. One is that they actually tend to have a substantial probability of fixation compared to neutral mutation. And when they do fix in a population, they can result in a similar sweep-like signature, but it's very, very restricted to a very small part of, like, to the target of fixation. So normally these won't matter at all, but in very, very low recombining regions, they could potentially look more like sweeps. So that's something to think about for low recombination regions. The second thing is that weakly deleterious mutations, as I said, also skew the site frequency spectrum more of the linked neutral alleles. And so again, that'll lead to issues with methods that use the site frequency spectrum to fit demographic models, etc. And the last one is that a lot of these weakly selected mutations, they can actually lead to this effect called associative overdominance. And all that means is that they sort of mimic the signatures of sort of balancing selection, increasing nucleotide diversity and LD in those regions. And so that's another thing to think of, another thing that like adds to trying to understand what's happening with how you predict genetic variation where there are these weakly selected mutations. And we also sort of found actually that if you have weakly selected mutations in populations, and you just take and you're looking at population differentiation approaches to look at SNPs that differentiate two populations, they can show up pretty highly as outliers. Only like, but if positive selection is weak, but if it's strong, then it's not a complication at all. So only when there is weak positive selection, they can actually sort of be part of the outliers and have confounding effects. So the second implication that I want to talk about is that it has been shown recently or a few times that you can take the best estimated demographic model, and if you simulate those and obtain those statistics, they may not always match the observed data. And yeah, I'm talking about the statistics that the model doesn't fit. Yeah, and so those things actually with ABC approaches, you can fit the statistic and you still might not explain it in the best model. And so, but what we found is that like one of the things that we think that so there are multiple reasons why that can happen, because you're overfitting a certain statistic in your model or your method, or maybe we just don't have the resolution to infer population history at that level. It could be another thing, which is that we're not accounting for some of the effects of deleterious mutations, et cetera. And so just as an example, we looked at what happens if you have deleterious mutations and a recombination rate heterogeneity in a Drosophila model. And if you just compare the one for strict neutrality and one that includes this information, the distribution of these statistics changes quite drastically. And so, it's possible that, if you don't account for these effects of selection and recombination rates, et cetera, you won't, you won't be able to explain how the data actually looks. And so we just think that what you want to start with is making sure you have a genome architecture that you're simulating that matches something realistic, and then building this sort of a baseline model where you account for heterogeneity of mutation and recombination rates and direct and indirect effects of purifying selection and population history, and hopefully data imperfections, like if you're masking the data and things like that. And so in closing, I just want to tell you that I talked about how if we don't account for these deleterious mutations, they can bias evolutionary inference. And that in general, because of that reason, we have to be cautious when we fit models to population genetic data. And our joint estimation framework represents sort of this starting point to construct a baseline model, accounting for these constantly operating evolutionary processes that are occurring in natural populations. And that can be used to assess the contribution of more episodic processes. And such a framework will allow the identification of many models in parameter spaces that are inconsistent with observations. And such sort of rejection of these scenarios can sometimes be pretty robust and more robust than the model fitting. So with that, I want to thank you for listening. And let me just quickly acknowledge my advisor, Jeff Jensen, and my very close collaborator, Brian Charlesworth, who've been part of all of the work that I presented today. My undergraduate students, actually, Helen and Emma, who are now doing PhDs in their own programs, and a bunch of other collaborators. And I just want to say that I am going to start at University of North Carolina, Chapel Hill in January, and I'm hiring. So please contact me if you want to work on things like these. Yes. Yeah, that's a very, I should repeat the question. So the question is, I didn't talk much about the ABC side of things and what statistics are helping us distinguish between selection and demography in this case. I think that's a very, very good question. And we try to look at that in the Drosophila paper. And I think the short answer is that a lot of LD statistics sort of were helping with the demographic history, and some of so mostly because we were also looking at it by region. So like really the functional regions are giving you more information about selection, and so is the recovery. Yeah, there's not a simple answer to that question. I think we ranked all the statistics based on how informative they were, and different. What we did learn is that it's good to have both LD-based statistics and SFS-based statistics to be able to make this inference. Yeah, we did do that. Yeah. Mm-hmm.
We have a question from Zoom. Silas asks, for human inference, maybe you want to just do it?
Okay. Oh. Yes. Okay. Yeah. Right. So He's asking for human DFE inference. The predictions for lethal mutation frequencies for your method were much lower. Do you think that could be an underestimate? Why or why not? Yeah, it's a great question. And I've also been thinking about that. I think there are a couple of different possible reasons for that. So one is that the other methods, I'm just going to go over them one after another, but so previous methods have sort of fitted a gamma distribution to the distribution of fitness effects. And I mean, gamma distributions have their own constraints because you're fitting 2 parameters. And so sometimes it is possible for because there's a constraint on the continuous distribution, it's possible that they were inferring a larger amount of lethal mutations than we have, but I don't know. The second reason is that, so a lot of the other methods, like the ones from Airwalker and Keatley that were there, those methods were performed on a set of genes that were like identified to be important for human health, or, so they have to be much more important. And so the lethal class was much stronger in those distributions. And so it's possible that it's like, because we have an arbitrary set, we're not actually going with the entire human genome. So it's possible that this set just happens to have fewer of those, or otherwise we're just not estimating. There's some error. So, yeah, it's possible that we just made an error in that lethal class. Yeah. Yes.
Thank you. That's a very nice talk. I want to expand on the one point that we had about there being clinic with the pattern of information. That sort of makes it really difficult to differentiate from analysis. Or was there some differences that in theory you might be able to parse those out?
Yeah, actually, oh, yeah, right. Okay, the question is that, is it actually, the effects I talked about, the associative overdominance effects, are they, is there a way to distinguish them from balancing selection effects? And yeah, I mean, the thing is, these effects are pretty small. They're not always that high. They're only high under certain conditions. They have to be very low recombining regions. They have to be, the dominance of mutations has to be less than, like, they have to be less than semi-dominant. There's very specific conditions. And there have to be many weakly deleterious mutations. If you actually have a distribution which doesn't have a huge number of weakly deleterious mutations there, you won't see those effects. So it's actually, you probably, I mean, if you see long haplotypes, they're probably, balancing selection, unless they're very low recombining regions, where I think there will be a problem. Yeah. 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 So the question is the question is, what is the expectation for the deleterious DFE to change across populations? Is that okay? Yeah. Yeah. I mean, so. Yeah, I mean, if you think about, for example, I mean, okay, so if you think about like the distribution of just the S or the selection coefficient, and if you were to, for example, think about like housekeeping genes or something, right? Like they shouldn't be very different from each other in different populations. But when you add in this effect of, if you look at it in terms of NES, which is, what's the strength of this selection in terms of, your population size, because how effective are they in that population? Then you can, you expect to see differences between populations, right? Because a larger population size, selection is more effective and is able to get rid of of more deleterious mutations and things like that. So, yeah. Yeah, so I don't know if there's a good answer to this question, actually, and I think people are trying to infer them in different populations so that we can try to get a better idea about this, this exact question. Yeah. Yeah, it's a good question. So the question is, could we have added recombination rate estimation in our joint inference procedure? Would that work? And yeah, I mean, it's a great question. We are kind of we're relying on the recombination rate estimates to be accurate to be able to, kind of to model this like recovery of background selection effects. So I mean, we didn't try it. So, I mean, I'm sure it could be done, but maybe you'll get, multiple posterior probabilities, which are possible in that scenario. Yeah. Hmm. Mm-hmm. Mm-hmm. Either, yeah. Yeah. I have not. Yeah, we have not. Yeah, but I'm sure that is something that's, I'm sure it's going to make a difference. Yeah. Has to be, yeah. But. Do you mean? Oh, yeah. Mm-hmm. Yeah. Oh, right. Yes, the question is, why was the time inference so short for the Yoruban population? No, that is also a good question. So the main thing is we didn't want to do too many parameters. So we had to restrict it, but, and we wanted to sort of focus on the recent population history because those were the, that's where background selection is likely to be possibly confounding it. So we wanted to test if that is really happening. And so we had to model a more recent change. Yeah. Mm-hmm. Mm-hmm. Yeah. Mm-hmm. Yeah, that's a good point. Thank you.